Hello and welcome to another of the eBiology Teacher Podcasts. I'm Mr. Doc, and today it's all about getting active. Specifically, we're talking about active transport, right? So in an earlier lecture, we talked about passive transport, that is the movement of materials into, out of, and within a cell without the cell actually having to use any energy, without the cell having to do anything. But in that lecture, we talked about there being some things that cells simply need to use energy to do. And that's where active transport comes in. So by the end of the lesson today, we should be able to explain the difference between active and passive transport, explain the difference between ion pumps and facilitated diffusion. We're going to examine why active transport is biologically important. And then there are going to be four new phrases or new terms that we're going to need to be able to describe. And they are processes, or processes, depending on where you're from. One's called endocytosis, pinocytosis, phagocytosis, and exocytosis. So, hello. We've got a lot to do today, but actually what I think is best to do is start with a quick review. And that is, when we talk about passive transport, cells need things, and we can get a lot of those real cheap, meaning that they're going to naturally move through the process of diffusion, keeping in mind that diffusion is the movement of materials from an area of high concentration to an area of low concentration. So in an example, we might consider watching a car or a truck or a matchbox car go down a hill, right? If I let go, I don't have to do anything, right? That happened on its own. That's a passive process. But if I wanted this thing to go this direction, I'm going to have to constantly add energy in order to get it to go against the gradient. Remember, in our previous lecture, we talked about this as being the concentration gradient. So passive transport will happen from an area of high concentration to an area of low concentration with no extra energy. But if you want to move in the other direction, well, energy is going to be required, right? So let's go ahead and take a look at our notes and see what we need to learn. Active transport refers to the idea that there are times when a cell must move materials from an area of low concentration to an area of high concentration. Again, that's against the natural concentration gradient. Remember, a concentration gradient is the direction in which materials naturally move. So that means against the gradient would be from low to high instead of high to low. So, active transport is when a cell actually uses energy to move materials around. So, active transport is when cells exert energy in order to transport materials either into, out of, or throughout the cell. Again, we think about this as sort of a uphill, downhill sort of thing. If I need something to move uphill, I'm going to have to use energy. It's just the way of it. Another reason why I might need to do that is if the item is just too big, like it can't fit through the membrane normally, so I'm going to have to figure out a way to fit it through. All right, so let's get a little bit more specific here. Cell membranes, one type of active transport is called ion pumps. Cell membranes have carrier proteins in them that act like doors to let materials in and out. This is similar to something like facilitated diffusion, but ion pumps require energy for a couple of reasons. The most important of which is cells need different concentrations of ions like potassium inside the cell and sodium outside the cell in order to function. Cells sometimes don't want equilibrium. They want to be imbalanced. So they would need to move things against the concentration gradient. Ions, or ion pumps, use energy produced from the creation of ATP, which we'll study more later, to force sodium out of the cell, even though there might be a greater concentration of sodium outside of the cell, while taking potassium in, even though there's already a higher concentration of potassium inside the cell. Again, they're going against the concentration gradient. One thing that's good to think about here is equilibrium. We talked about equilibrium being where everything balances. Well, ion pumps fight balance. They don't want the cell to be at equilibrium, and your cells function better when there is an imbalance. So we need those ion pumps. Let me show you an example of this that I think will help. Okay, this is an example of an ion pump. Let's take a look.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient.
So now you can see here there are a lot of sodiums outside the cell and a lot of potassiums inside the cell, but that's what the cell wants. And naturally, potassium would want to go this direction, go out of the cell, and sodium would naturally want to move inside the cell. And the sodium-potassium pump keeps that imbalance. It keeps that imbalance from preventing it from reaching equilibrium. This is incredibly important in us with our nervous system. The way our nerve cells work, they have to have an imbalance. They have to have a sodium-potassium imbalance in order to work. So sodium-potassium pumps have to use energy. You saw that ATP kind of attach. Basically, and again, we'll get into this more later, that ATP is how we're spending energy. That takes a little energy away from us in order to make that happen. Okay? So let's take a look at our next page of notes. Vacuoles, which we talked about earlier when we talked about lysosomes, are little membrane-bound sacs. So let's say that some molecules are so big they can't even pass through the cell membrane, not even through carrier proteins. In other words, there ain't a door big enough, right? Some molecules like proteins and carbohydrates are required for cells to live, but they can't easily fit through the membrane, and there's no real transport protein that is going to help them out, or no carrier protein that's going to help them out. Cells can use their own membranes, their outer membranes, to form vacuoles or vesicles, little sacs, and bring the sacs themselves into the cell. This means that the fluid mosaic model of membranes, is really super important. You see, the membrane can change shape a lot. The membrane can come in on itself and reconnect. So that means that the vacuoles are made out of the same stuff as the outer membrane. The membranes inside those organelles are just like the one on the outside. So they're easily exchanged. Their little pieces can just change from a vacuole to being part of the cell and back and forth. So this means that there are a couple of different ways we can move in these really large molecules, either by sucking them in or spitting them out. And let me show you how that works. Okay? Endocytosis is the process of taking in food through these little end folds, okay, these little fold ends or pockets in the membrane. Exocytosis is the opposite. It just takes a vacuole, fuses it to the outer membrane, attaches it to the outer membrane, and then releases the stuff into the outer world, right? So if we look here, this endocytosis, this food touched the membrane, and the membrane kind of caved in on itself. The way I like to think about that is Scooby-Doo. You know, when Scooby-Doo leans against a bookcase, and then it's like, woo, right? It just falls right back in. That's kind of the way endocytosis happens. If a cell detects something it wants, it just kind of goes, makes a little hole, falls in the hole, closes around on itself. It's kind of a cool thing. Exocytosis, on the other hand, is where that membrane touches the outer membrane from the inside, and then the vacuole then becomes part of the membrane itself. All right? Now, pinocytosis is a special kind of endocytosis in that it's bringing in fluid. Okay, it's similar to endocytosis, except it's the forming of the vacuole to take in like big boluses or big collections of fluid. Phagocytosis is when the cell basically goes out and gets something. Okay, so I'm going to show you a little bit about this in a second. So a phagocytosis is when a cell creates kind of extensions and reaches around it. So when we think of endocytosis, this is me being stupid. I think of Scooby-Doo falling through a bookcase. You know, he leans against it, and this bookcase just sucks him in and he falls. Phagocytosis is like the bookcase moving and the bad guy reaching in, grabbing him, and pulling him in. Okay? I think I have an animation that explains it much better than I do, clearly. All right, let's take a look at this.
The substances taken in by single-celled organisms are often particles or large polar molecules that cannot cross the hydrophobic plasma membrane. Many single-celled eukaryotes employ endocytosis to ingest such food particles. In this process, the plasma membrane extends outward and surrounds the food particle. Cells use three major types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterium or a fragment of organic matter, the process is called phagocytosis. If the material the cell takes in is liquid, it is called pinocytosis. Specific molecules, such as low-density lipoproteins, LDL, are often transported into eukaryotic cells through receptor-mediated endocytosis. Molecules to be transported first bind to specific receptors on the plasma membrane. The interior portion of the receptor protein is embedded in the membrane. The protein clathrin coats the inside of the membrane in the area of the pit. When an appropriate collection of molecules gathers in the coated pit, the pit deepens and seals off to form a coated vesicle, which carries the molecules into the cell. Exocytosis is the reverse of endocytosis. This process results in the discharge of material from vesicles at the cell surface to the outside of the cell.
Let's take a look at that again, okay? And I want you to notice how the membrane of the vesicle or the vacuole either started out as part of the cell membrane or becomes part of the cell membrane. Take a look at this, and I'm just going to use the rewind feature here. But if I pull this back a little bit, see how the membrane of the vacuole, that's this thing right here, as it approaches the cell membrane, it just becomes part of it. And then when it spits out, the vesicle is gone. That's because this part of the membrane is now the vesicle. The same is true with the endocytosis. Keep in mind that when this bacteria touches, the cell reaches around it, and this portion of the cell membrane, which is still part of the outside cell membrane, now becomes the vacuole, right? So this portion here used to be part of the cell membrane, but now it's its own vacuole. And by the way, this little thing here, this little vesicle, would become a lysosome now. Because it's got the bacteria in there, it's going to start secreting enzymes, break that bacteria down. So that's how that works. Now, phagocytosis is most well known because it's used by something called amoebas. Now, amoebas actually eat their prey using phagocytosis, and I have a micrograph video. So this is a video taken through a microscope of several amoeba eating their dinner, and it's really kind of freaky. It's kind of like a horror movie. Check this out, right? This is the amoeba. Notice how it's reaching around, right? It's reaching around that little food. It looks like a paramecium, maybe a dead paramecium or two. It's going to envelop. It's going to reach around. It's going to grab it, and it's going to form a vacuole. Now, once these two little pseudopods, that's what these extensions are called, meet, we now have a vacuole. That's now a vacuole, or what will become a lysosome, and those two guys, unfortunately, are not going to make it, right? So I'll show you another one here. See, there it is. I'm going to eat ya, right? And now we've got a little vacuole. Here it is again. It actually, that one looks like Pac-Man. Right? So this little vacuole here contains what that amoeba just ate. And now that amoeba is going to digest that and use that for itself. Right? That's what it's going to happen. So, as you can see here, though, because we're changing the shape of the membrane, I mean, we're basically changing the shape of the cell. That's obviously going to require energy. That makes sense, right? So, these types of energy, or these types of transport, will require the cell to spend a little capital. It's going to spend a little energy. All right? So, let's take a look at our objectives. Explain the difference between active and passive transport. I think the easiest way to do this is to use the hill analogy, okay? Active transport requires us to push or pull things through, right? It requires us to go against the way things would naturally move. Passive transport doesn't. Passive transport requires no additional energy. Things are going to move from an area of high concentration to a low concentration, just like a car going downhill, right? So passive transport, no energy spent. Active transport, energy spent. Okay? Now, what about the difference between ion pumps and facilitated diffusion? What is the difference? Well, keep this in mind. Remember, they both use carrier proteins. They both have little membranes little proteins in our membrane, and they're very specific, right? A carrier protein can only transport the things that carrier protein fits, kind of like the whole enzyme thing, right? It only fits the right way. So, the deal here is when facilitated diffusion occurs, there's no energy expended. Things are going from an area of high concentration to an area of low concentration. They're going downhill. So it's still a passive process. The biggest difference is ion pumps work against the concentration gradient. They're going uphill, right? They are moving things from an area of low concentration to an area of high concentration, and that's going to require some energy. That's going to require you to spend some energy. So the biggest difference between sodium-potassium pumps or ion pumps and, say, facilitated diffusion is the direction they're moving stuff. Facilitated diffusion is passive and moves stuff with the concentration gradient. Ion pumps is active, uses energy, and moves stuff against the concentration gradient. So why is this important? Why is active transport important to cells and biology in general? Well, that one should be easy. We need stuff in order to survive. We need proteins, we need lipids, we need nucleic acids, and most of us get that stuff by taking it from somebody else. That stuff's too big to get into our cells, so we have to move it. It takes some energy to move it in, right? It's biologically important because without it, we wouldn't be able to survive. We wouldn't be able to get the things we need into our cells. Okay? Now, the last part here, describe the process of endocytosis, pinocytosis, phagocytosis, and exocytosis. Well, endocytosis actually is a kind of a broad range of things. Endocytosis is any of those processes that bring things into the cell. The two that we talked about are pinocytosis and phagocytosis. The video also talked about receptor-mediated endocytosis, and that's just kind of where things fit into a protein that's on the surface of the membrane and it sucks it in. Phagocytosis, on the other hand, looks something, well, let's say like this, right? Oh no, don't get me, right? Here we go. This guy's coming out. He's going to move in and he's going to eat this guy. See how he's reaching out, he's grabbing him, right? So phagocytosis might look something like that. Whereas pinocytosis would look similar to that, except it wouldn't be solid, it'd be liquid, right? Now, exocytosis would probably look something like this. Exocytosis would look something like this. See how we have this vesicle here? And that thing is going to go to the edge, and it's just going to spit it out. That's exocytosis, is spitting it out. And by the way, while I'm at it, I have a pretty neat sort of diagram of an ion pump that I think might help you here. See how we have a whole bunch of potassium on the inside and a whole bunch of sodium on the outside, but it looks here like potassium's being moved in and sodium's being moved out against the gradient. There you go. That's an ion pump. All right. So those are the differences between those ideas. I really hope this helped, and I hope you are able to distinguish between passive transport, those things like osmosis, diffusion, and facilitated diffusion, where things are just kind of going downhill, no energy, and the things where the cell actually, oh, I'm frozen, where the cell actually has to do something in order to accomplish what it wants to accomplish. All right? Visit the website if you need more information or ask me in class. I look forward to talking to you. Bye.